Okay, folks, so this video is going to focus on basically one topic, okay? And that topic is known as oxygen hemoglobin affinity. And sometimes students overcomplicate this topic. I know I did when I was learning it for the first time. Um, I really struggled with this graph here. This is called the oxygen hemoglobin dissociation curve. And sometimes that just overcomplicates things. So if you don't quite understand this graph, it's not the end of the world. And this topic is actually fairly straightforward. It just brings in a few things that you have to remember from, say, biology, such as cellular respiration. What do we need for that, and what is the result of that? And we'll get to that in a minute. But let's look at some of the words we're going to see here. So, oxygen hemoglobin affinity. So, affinity is the tightness. All right. So, hemoglobin obviously transports oxygen, but the affinity it has for oxygen needs to be able to change. Okay. You can't have hemoglobin grab oxygen so tightly that it can't let go of it, right? Because hemoglobin is a transporter, right? It's going to have to grab onto oxygen in the pulmonary capillaries, but then when it gets to the systemic capillaries, it needs to let go of it, right? So that oxygen can then diffuse into cells. So this whole concept revolves around the idea that something has to change hemoglobin so it knows that it needs to let go of oxygen, okay? And that's called oxygen hemoglobin affinity. Now, the graph just describes the relationship between how much oxygen there is and how much you can saturate the blood, all right? So how much can you get into that blood? And let's go over the curve first. And again, it'll be okay if you don't quite understand the curve, but it does help in some cases. So the black line represents kind of your normal hemoglobin, all right? And if you look here at the 40, that's a certain amount of oxygen, all right? So 40 millimeters mercury of oxygen. And if hemoglobin is in this normal state, it can be used to saturate about 73%. All right, so that's kind of a normal situation. If we change hemoglobin, all right, we can either get more oxygen transporting or we can lower the oxygen transportation or saturation. So notice when the curve shifts over to the right, what that does is it means that you can't saturate at the same level as you did with the black curve. So what that means is something must have decreased the affinity hemoglobin has for oxygen. So that tightness decreased, all right? It weakened the attraction, if you will, all right? And what that does is it promotes more of the unloading of oxygen, all right? So that's going to cause hemoglobin to let go of the oxygen instead of hold on to it tightly. If this curve shifts to the left, notice all of a sudden for the same amount of oxygen, you can get more of it saturating the blood. So what that means, something must have happened that tightened the affinity or increased the affinity. So that's how you look at this graph. You don't really, I don't think it matters whether you look at the numbers or anything. You just look at what causes it to shift to the right versus the left. If it shifts it to the right, that means the affinity decreases and hemoglobin will release oxygen. If you can shift it to the left, it'll hold tighter to the oxygen. Now notice there's certain things, there's certain conditions, there's certain factors that will shift this curve, all right? The ones that shift the curve to the right are going to lower the affinity. So that is an increase in carbon dioxide levels, a decrease in pH, so that means the area is more acidic, and an increase in temperature. All right. What shifts it to the left is the opposite. Less carbon dioxide, higher pH, lower temperature. So what does this mean? let's channel our inner hemoglobin. So I want you to think about you being hemoglobin. Okay, what do you do? Well, what are you? For one, you're largely protein, right? You're the globin. You've got four of those. You've got four hemes, right? And hopefully you've got four irons, all right? And you're going to enter the pulmonary capillary. Well, remember that pulmonary capillary is where you're supposed to be, grabbing oxygen from the lungs, okay? So you want a high affinity, okay? So you want, lower carbon dioxide, slightly higher pH, and lower temperature, all right? So you grab onto that oxygen very tightly. Then where do you go? Well, let's do a little cardiovascular review. If you're in the pulmonary capillaries, you're becoming oxygenated, right? You're grabbing as much of that oxygen as you possibly can. So the blood has become oxygenated. We got to go back to the heart. So we're going to go pulmonary venules and veins. Then we go to the left side of the heart, left atrium, left ventricle. Then we're pumped out, right? Now our job is to go deliver. So we're going to go aorta. We're going to go systemic artery, systemic arterial. And now we're going to enter a capillary, systemic capillary. Now what happens there? Well, we're supposed to deliver, right? So if you're hemoglobin, you now have to lower your affinity, right? You can't be holding on to oxygen so tightly because then it won't be able to release and diffuse into the cells. So we have to have conditions or factors that decrease the affinity. So what happens is you go from over here, you shift the curve over. So tissue at systemic capillaries, if it has higher carbon dioxide, if it's slightly acidic and higher temperature, that's going to weaken the affinity, lowers the affinity, and it causes hemoglobin to unload or release the oxygen. And that should make sense. So now let's go back to this idea of cellular respiration. If you're a tissue, right, your muscle tissue, for example, and you're very active, let's say you're running and you've got quadricep muscles that are contracting and relaxing, those cells have to be generating a lot of ATP. So as a result of generating a lot of ATP, you also generate a lot of carbon dioxide, okay? You also become acidic. Now, one reason you become acidic is the carbon dioxide, but you also generate some other kind of byproducts, all right, that are acidic. And then you become warmer, right? Another byproduct or result of cellular respiration is you produce heat. So that tells hemoglobin, okay, if this tissue is highly metabolic, right, it's producing a lot of energy, it's generating more of this, I need to lower my affinity because if tissue is generating a lot of ATP, it's going to require more oxygen, okay? So that's the big picture here. We need to get hemoglobin to grab on tightly and then let go, okay? So I think of hemoglobin kind of like a magnet, right? But it's a magnet that you can kind of alter how strong it is. You want it very strong until it gets to systemic capillaries. But now think about that same muscle if it's no longer contracting and relaxing. So let's say you're just sitting, in a chair or you're sleeping. That same muscle is not generating as much ATP. So it's not generating as much carbon dioxide, it's not becoming acidic, and it's not warm. So that's going to tell the same hemoglobin molecule that, this tissue doesn't require as much oxygen because it's not as active. So the affinity won't decrease as much. So we don't deliver as much oxygen. All right. So to me, this is one of the more interesting concepts in this chapter. But again, it requires you to understand a lot of different things. Cellular respiration, a little bit of pH, right? What is high versus low pH? Low pH is acidity. Higher pH is, either less acidic or even basic. So it requires you to bring in some of your scientific knowledge. But once you just get the idea that, yeah, it makes sense. You can't have hemoglobin just grab oxygen and never let go. You need to somehow get it to lower its affinity so it can release that oxygen. And then there's variations of that, right? Depending on how much oxygen that tissue needs. Okay, so the big four: acidity, carbon dioxide, temperature. We just mentioned that here. There's one more thing. It's a molecule, which we'll talk about in a subsequent slide. So we're going to kind of go over each one of these separately. All right. So this next slide focuses just on the pH angle. Same kind of idea. We're looking at, normal situation shifting to the right. What does that mean? Well, that means it's going to lower the affinity. We see that if we drop the pH a little bit, that's going to lower the affinity. Now, what is actually doing that? Well, you have to understand what acidity means. Acidity means you're generating more of these hydrogen ions. All right, that's the measure of pH, by the way. The more of these you have, the lower the pH. All right, the lower the pH, the more acidic you are. And it's actually this hydrogen ion that ends up binding to hemoglobin and altering its structure. That's how you affect the affinity, by the way. If you change the structure of hemoglobin, you can lower its affinity. Hemoglobin's a protein, and it's all about the shape of protein. When cells make a protein has to fold, and it's that folded structure that allows the protein to function. Well, if you alter that folded structure, it alters the function. In this case, it goes from a higher affinity when this is not attached to a lower affinity when it does attach. All right? Now, what generates this hydrogen ion? Well, it could be lactic acid that is produced. It could be carbonic acid. Those are the two main acids that are produced when you have metabolically active tissues, meaning it's a tissue that's generating more energy. Okay? This whole concept has a name. It's called the Bohr effect. All right? So the Bohr effect is when you have a low pH, aka acidity, which forms hydrogen ions, also known as protons, and that alters the affinity. All right? It lowers the affinity. All right? So if you were to just take hemoglobin and it's got oxygen on it, and you just change the pH, it'll let go of that oxygen if you lower the pH. If you raise the pH, it'll hold on to that oxygen. Okay? Next one is the carbon dioxide angle. And again, it's very similar. It's just now instead of the hydrogen ion that binds to hemoglobin, carbon dioxide can bind to hemoglobin. It actually binds to the globin portion. And when you have more carbon dioxide, all right, because the tissue is more metabolic, it's going to alter and lower the affinity. All right, so those two go hand in hand. All right, low pH and high carbon dioxide. And carbon dioxide is actually one reason why you get that hydrogen ion, because it undergoes this chemical reaction. Most important chemical reaction you're going to learn in this class. When you have carbon dioxide, it will react with water, and there's an enzyme that facilitates this called carbonic anhydrase, and you will get an acid. So the more carbon dioxide you have, the more acidic you will be. It just so happens this is also how we transport carbon dioxide, but this is an acid, all right? And it plays a role in lowering the affinity. All right, next one was the temperature. Temperature also alters the structure of protein, all right? So when you raise the temperature a little bit, that has a very similar effect on hemoglobin. Alters the shape, lowers the affinity, unloads more oxygen, all right? So tissue that's more active tends to be warmer, okay? All right, the last two things. This BPG molecule that I mentioned a couple slides ago. The fancy name is 2,3-bis-phosphoglycerate, and it's a molecule inside red blood cells that's produced when the body is generating certain hormones. Those hormones are thyroid hormones, human growth hormone, epinephrine, norepinephrine, and testosterone. So think about when your body's generating these. Basically, if you're generating more thyroid hormones, your metabolism has gone up. If you're generating growth hormone, you need more metabolism because you need energy to grow. If you're generating epinephrine and norepinephrine, you might be in a fight or flight situation where you need more energy. And testosterone stimulates growth. So in all these cases, you need more energy. You need more ATP. So if you need more ATP, hemoglobin needs to know to release more oxygen into those tissues. All right, so it's all about telling hemoglobin, you got to lower your affinity and get more of that oxygen into the cells. All right, and then the last thing is just a comparison. There's actually two forms of hemoglobin. Fetal hemoglobin, which obviously is the one that a fetus produces, versus adult. So after a fetus is born, we actually change the type of hemoglobin we make. So think about what a fetus has to do and what mom's blood has to do. All right? Mom's blood enters a placenta, so does the fetal blood. Well, there's two hemoglobins there. You don't want them fighting over the oxygen. You actually want oxygen to diffuse and get into fetal blood instead of just staying in mom's blood. So it's almost like you've got two magnets, but one's a little stronger, right? The fetal hemoglobin is a stronger magnet for oxygen. So oxygen, therefore, will diffuse out of the maternal blood and get drawn into the fetal blood. All right? So if you're ever asked, fetal hemoglobin has a higher affinity for oxygen than adult hemoglobin. Or, don't forget, I could ask you the reverse. Adult hemoglobin has a lower affinity than fetal hemoglobin. All right? And that's how we can transfer oxygen from the maternal blood, mom's blood, into fetal circulation. All right, so we're going to end that video. One topic. I think it's a really interesting topic. I think it's a very important topic. So we're going to start the last video using a couple figures that are going to be kind of a review. All right, so this slide is kind of a review. The next slide is going to be a review. And then the last topic in the next video is going to be how our nervous system regulates our breathing. So how do we increase our breathing rate? How might we decrease our breathing rate?